Okay, so we have talked about genetic diseases, and now we're going to move on to some crosses that have like really different alleles than what you're used to seeing. So the first thing that we're going to talk about is like blood type, right? So blood type is considered to be codominant. And so something is codominant when both alleles produce an associated phenotype equally. So blood type is one of those examples of a codominant trait. And so if you've probably heard something like, oh, type A, type B, or type O, and what they're actually talking about, the A, the B, those are glycoproteins that the red blood cells use to mark them as like self. So your body does distinguish between itself and other cells, and one of the ways that it does that is to use antigens to mark its cells. And so in this case, these are just proteins with some sugars on them. The genes that are associated with this put those sugars on the proteins of the red blood cells. And so that's how we end up with a blood type. So there are two antigens that could be produced. So those are, again, just proteins with sugars on them that sit in the membrane, and they will either display an A antigen, which is that glycoprotein, a B antigen, which is a different glycoprotein, or you might have no antigen at all. And so any combination of these three alleles can occur, which is how we end up with our different blood types, right? So if you have type A blood, it means that you have the type A glycoprotein on the surface of your red blood cells. If you have type B, it means you only have the type B version of that glycoprotein on your red blood cells. You can also be AB, and this is where the codominant comes in. Because both of those glycoproteins are present, they are expressed in equal amounts. This is really where we're calling it codominant, because they are equally type A and they equally have the type B glycoprotein on their surface. Or you can also be type O, where you don't have any antigens on the surface of your cells at all. So there are 4 official blood types: type A, type B, type AB, and type O. So in this case, we're changing how we use our alleles. Really? Hang on, let me get this out of the way. Okay. Our alleles are always going to be represented by a capital I and a superscript for either A or B. So for type A, did I talk about this? Yeah. Okay. And then there's type O, which is represented by the lowercase I. So this one is unique. If someone is type O, you automatically know their genotype is little I, little I. And we're calling this kind of recessive because it's just the presence or absence of an antigen. So for type A, there are 2 genotypes that can produce a type A phenotype. One of them is the homozygous, where you're homozygous for IAIA, you'll have those type A glycoproteins. Or you only have one copy, you're heterozygous for it. So if somebody says you're heterozygous for type A, it means you have one uppercase I with a superscript A and one lowercase I. So this is a heterozygote. Phenotypically, they look the same. In both cases, the cell has those type A antigens on its surface. Type B works the same way. I can either be a homozygous type B or heterozygous type B. That should be an uppercase. I keep thinking I'd change that. But either one will produce a type B phenotype. So type AB and type O are unique in that they have only one genotype associated with them. So if you're type AB, you have one A allele and one B allele. And if you're type O, you only have the recessive allele. So I am type O, so I have both lowercase I. And so when you're looking at the problems, these are you wish that these happen because you automatically know the genotype for each. It's only the type A and the type B that could be either one. So when we're setting up these crosses, so let's say that we want to cross an AB, someone who's type A, B against another person who's type AB. So we set up a cross like this, because if they're type AB, it can only be one genotype. And again, you have to have the I here with the superscript A. If you write just A and B, that's incorrect. You must have the uppercase I with the superscripts A and B. So okay, I've got my two type AB individuals, and the crosses work the same way. You just bring, this allele down the row and this one down the column. So this first one would be IA, IA. This one would be IB, IA. So this would be a type AB. This one would be a type A. And this one, again, would be another type AB. And then this person would only be type B, because that's the alleles that they have. So these would be your ending genotypes and phenotypes for this example. So let's say I have a cross where I'm saying I want to cross somebody who's heterozygous with type A with somebody who's heterozygous with type B. So remember, heterozygous in this case means that you have the mismatched alleles. So I have the I uppercase A for my heterozygous type A with one recessive allele here, and I'm going to cross that person with a type B allele, and because they are heterozygous, their other allele would be a lowercase I. So when I complete this cross, I'll have one individual who's type AB, one individual who is heterozygous type B, one individual who is heterozygous type A, and one individual who is type O. So this is a relatively special cross. You see a lot on the exams. When you cross the heterozygotes for type A and type B, you end up with every ending possible genotype and phenotype. So that's the 411 on blood types. So we have been focusing exclusively on autosomal traits and diseases, but now we're going to talk about diseases that are linked to the X chromosome. So the X chromosome is somewhat large. There are around 800 genes on them that range in function from anything from your neurons to how we form blood to clotting and photoreceptors. So just a reminder that females have 2 X chromosomes, so we are diploid for all of these genes. This is unlike males who only have 1. So for diseases that we find on the X chromosome, because men only have 1, that makes men more vulnerable to all of these X linked diseases, because again, they only have 1, and so if something goes wrong in that copy, they have the disease. So one of the diseases we're going to talk about is X-linked red-green color blindness. So this is a mutation in either the OPN1LW or OPN1MW, which is an opsin pigment. So this protein codes for, so this protein either absorbs yellow or red light, and it allows you to discriminate between them. So this is on the X chromosome, so this gene is X-linked, and like most mutations, this one is caused by recessive mutations. The mutations like break the copy, so you would need 2 hits if you're female, but only 1 if you're male, right? So this is what it actually looks like. So somebody who has color blindness can't really distinguish between the reds and the greens. It all sort of looks the same to them. So in this case, the alleles are specific too. They're not, big B or little b. You have to indicate that they are on the X chromosome so that you're demonstrating that you understand this is an X-linked disease. So the alleles we'll use are X superscript B, big B, or X superscript little b. So the big B is a dominant allele. That means that you're healthy. So in this case, we have a female non-carrier who is not colorblind. And in this example where we have the mixed alleles, this is a carrier, so she is female because she has 2 X's. So this is a female carrier who is also not colorblind, because you need 2 small copies if you're female to be colorblind, like this person here. So XX means you're female, and having both recessive disease alleles means that this female is colorblind. So we'll do it separately for the males. In this case, there's only 2 options. A male is always XY. If the male has the dominant allele, they're not colorblind, they're fine. But if they do have that smaller allele, they do have red-green color blindness. So males are going to be affected more often for all of the X-linked diseases. So what is happening with color blindness, like usual, when you make mutations in that gene, it ends up with mutations in the protein that can then not fold or function correctly, and so red and green cannot be distinguished anymore. So now we're going to set up our crosses, and we're going to use the XY nomenclature, and so it is going to matter, who's who. So we have a male with color blindness. So male, first of all, is going to be XY. And if he has color blindness, it means that he has the lowercase allele. So we're going to put that there. And then if we have a female who's a carrier, remember carrier means that you're heterozygous, and if you're female, you are XX. So this female is going to be heterozygous for the colorblind allele, where one of them is the dominant normal copy and one of them is a disease allele. And so when we do our cross, we have one girl who is heterozygous, one female who has both disease alleles, so this means that she's going to be colorblind. And let's see, one male who has normal vision and another male who also has color blindness. So when you're setting these up and you're kind of reporting, give the distinction between the sons and the daughters, because you can't lump them all together anymore. So you have to like split it, say 50% of the daughters are colorblind, or and 50% of the daughters are carriers. In this case, 50% of the sons have normal vision and 50% of the sons are colorblind. So it changes a little bit how you report the genotypes and phenotypes. The last disease that we're going to talk about is hemophilia. So this is controlled by a gene called factor VIII, so coagulation factor number 8. And what this protein does is it signals for a blood clot to form. And so this is found on the X chromosome, so it is going to be an X-linked disease. And again, like most of them, it's a single base mutation, so this one is going to be X-linked recessive. The single base mutation leads to just a non-functioning protein, right? So if that protein is broken and it can't signal anymore, you basically never form blood clots. And so this leads to uncontrolled bleeding upon injury. People who have hemophilia have to get blood transfusions from healthy blood so that they can get that factor VIII in there. If they do not receive treatment, they mostly die before they're 10 years of age, which is really sad. So we're using the same nomenclature because this is an X-linked disease where we're using our X's and our Y's, so we have to distinguish between our males and females. So okay, we're going to start with a female with hemophilia. So she's recessive. She's homozygous recessive. So here we have our female who has 2 small copies of the hemophilia disease gene, and then we have a normal male, so he is XY with a copy of the dominant allele. So we'll go ahead and set that up. We've got our male XY with the dominant allele. We've got our female over here who is XX, and she has hemophilia, so she has 2 bad copies of that hemophilia gene. And we'll go ahead and do our cross. And so we've got 100% females who are carriers. Remember, we don't call these just heterozygous, they are now carriers. And for the boys, we have 1 person at least who is affected. No, we've got 2 of them. So 100% of the sons have hemophilia and 100% of the daughters are carriers. But there are 0 who have actual hemophilia. So that concludes our genetic diseases. So stay tuned till next time.